Welcome to Circuit Buddy. Today we will be discussing about cell membrane. Cell membrane is a very important topic of class 11 biology. It is also important for Neet. Okay, so let us discuss about cell membrane in details. Let's proceed. Keep watching the video till the end so that you can understand and remember all the portions or important points of cell membrane. Okay, so discovery. First proposal of the presence of a lipid bilayer was made by Grenell and Gotter in 1925. Okay, so after that, Davison and Danieli proposed that plasma membrane was composed of a lipid bilayer which was lined on both its inner and outer surface by a layer of globular proteins. So what does it mean? It means that in the Danieli-Davison model, which is called the sandwich model also, okay, so these are the lipid molecules, okay, and they are arranged in two layers, and these layers are called leaflets. Okay, so these layers are called leaflets, and you see that these are the non-polar tails of the lipid molecules. This is the polar head regions of the lipid molecules, and one more thing is that these are the globular proteins, okay, in between which the lipid molecules are sandwiched. Okay, so that's why we call it a sandwich model. This is also called a PLP model, okay, or the Danieli-Davison trilaminar sandwich model, and it was proposed in 1935. Later it was revised in 1950s due to some problems regarding the passage of solids through the plasma membrane. Okay, so after that comes the fluid mosaic model. So why is it named fluid mosaic model? First of all, it was proposed by Singer and Nicolson, okay, in 1972. Okay, and it is called fluid mosaic model because 1972, and fluid refers to the dynamic nature of the cell membrane. Okay, dynamic means the lipid molecules can move from one place to another. Okay, so that's why we call it a dynamic system, okay, a fluid state. Now mosaic, why we call it a mosaic model? Mosaic means the protein molecules are embedded within the lipid bilayer like mosaic. So you can see these are the protein molecules embedded within the lipid bilayer like mosaic. So we call it a fluid mosaic model. Okay, now you can see that here in this structure, these are the head groups of the lipid molecules, these are the tail groups that are there, and they are arranged in two leaflets. Okay, so that's why we call it a lipid bilayer. Okay, and these are the protein molecules. Some are transmembrane proteins. So this is an example of transmembrane protein. Transmembrane proteins are those proteins which actually pass through the cell membrane all the way. Okay, so it can pass all the way through the cell membrane. So it is called a transmembrane protein, a transmembrane, okay, transmembrane protein. And other protein molecules are called the peripheral proteins, which are actually present in the periphery of the cell membrane. There are other molecules also that are attached to the cell membrane. We will be talking about them later in the session. We will be gradually discussing about various types of molecules, the membrane lipids, the membrane carbohydrates, and the membrane proteins. Okay, so keep watching the video to clear your doubts. First of all, let us take a membrane lipid. So this is a membrane lipid. We call it a phospholipid because the phosphate group is attached to the lipid moiety, and this is the glycerol backbone, okay, to which two fatty acids are linked. Okay, so this is actually the structure of a phospholipid. So phospholipid means and phosphate group is present to it, and as it has a backbone of glycerol, so we call it phosphoglycerides. Okay, so as it has the phosphate group, it has polar head group. Okay, so phosphate is polar, so it has a polar head. Okay, so polar head group and a non-polar tail region. Okay, so that's why we call it amphipathic. Amphi means both, so amphipathic, amphi means both, okay, and pathos means region. So both regions are present in the lipid molecule. Okay, so hydrophilic region and hydrophobic region, both regions are there, so we call it amphipathic molecules. Now phosphoglycerides I have told you earlier, so I am not talking about them now. So there are various types of phosphoglycerides. What are they? Okay, so according to the various types of R groups present, okay, we can name them differently. Okay, so you can see that it is the generalized structure of a phospholipid to which an R group is linked. Here it is choline, that's why we call it phosphatidylcholine. Okay, so this is the structure of choline. If there is another group which is attached to the phosphate group, then we would name it differently. So here in this structure you see there is an ethanol amine, so that's why we call it phosphatidyl ethanol and the rest of the structure is that of a phospholipid moiety. Okay, so that's all. Now after that I want to talk to you about the phosphatidyl ethanol. It is a very important phospholipid. Actually phosphatidyl ethanol actually promotes the curvature of the cell membrane. It is present in the inner side of the cell membrane. We will be talking about them more. Okay, so later in this video, keep watching the video till the end. Now it is phosphatidyl serine. If you don't know what serine is, please watch my videos on protein biochemistry. There I have mentioned the amino acids, how to draw them, how to remember their structures. Okay, so this is the structure of serine which is attached to the phosphate group here, and this is the generalized structure of a phospholipid. So that's why we call it phosphatidyl serine. Okay, so these are the various types of membrane lipids that are present. There is another type of phospholipid, sphingolipid. Okay, that is, or membrane lipid, that is sphingolipid. So actually here no glycerol backbone is there. Okay, there is actually a sphingosine backbone. Okay, and to which you can see there is a choline attached to it, attached to the phosphate group, and to the amino group there is a fatty acyl chain. Okay, so we call it sphingomyelin. Sphingomyelin is also very important. Sphingomyelin is present in the outer liblet of the cell membrane. We will be talking about them later. Now the lipid bilayer is asymmetrical. Why I am telling you this? Because asymmetry refers to the different types of composition of the lipid bilayers, liplets. Okay, so lipid bilayer, we can say this. So there are two liplets, the outer liblet and the inner liblet. The outer liblet has a different type of composition. Okay, and it is different from the inner liblet in terms of composition. The outer liblet has two types of phospholipids the most. Okay, so we can call it coma in short. When a person is in coma, he is out of the real world. So out means outer monolayer. We can remember this in this way. So C means phosphatidylcholine and M means sphingomyelin. So these two phospholipids are present in the outer monolayer the most, and here you can see another word, that is exercise. In this lockdown situation, actually we cannot go outside, but we have to keep ourselves fit, and for this we have to exercise. So we have to stay inside. Inside means inner monolayer. So you can memorize this in this way. So E for phosphatidyl ethanolamine, and you have the I for the phosphatidyl inositol, and then you have the S means phosphatidyl serine. Okay, so these phospholipids or membrane lipids are present in the inner monolayer. Okay, or we can say the inner liplet. Okay, so the lipids can change their position laterally very easily but they cannot easily change their location from one leaflet to another. For this, they have a specialized enzyme which I call the membrane translocator. Okay, so the membrane translocator is a very important enzyme. Translocator, let me write it here. Translocator. Okay, so the membrane translocator is an enzyme which actually helps in the flip-flop of the lipid molecules from one leaflet to another, and when a particular type of phospholipid is present in a wrong leaflet, it actually determines the destruction of the cell membrane or maybe blood coagulation. Okay, so this is an important characteristic of the cell membrane, and composition of the cell membrane is very important to be remembered. Okay, so thank you very much for listening to my video lecture. I will be coming soon with another part of the video. Okay, thank you very much.